So thinking about retrobiosynthesis differently, its name is of origin from retrosynthesis, an approach that should be familiar to any of you that have taken organic chemistry. In retrosynthesis, as you might recall, you would have these examples where you have a particular product here that you might have or want, and you have to figure out how from certain reagents in the stockroom you can get there from a starting molecule. And sometimes you'll have this posed differently, where instead of showing you what it's starting from, this is just an image from a homework assignment from a different class, where you might have these different modes of organic chemical reactions, like a backside nucleophilic attack or any of these other mechanisms. And so you've probably had some practice trying to figure out how you get to some organic molecule from some reagents in the stockroom. Well, retrobiosynthesis is like that in some respects, but also quite different. Because, instead of something like a Grignard reagent or particular like, haloalkyne or what have you, you have just all these enzymes, and you have a naturally occurring, instead of thinking about the stockroom, you have naturally occurring metabolism, and that's kind of your stockroom. But more likely, you have a more narrow stockroom, which is the metabolism of your organism, the one you want to engineer. So because so much of this is dependent on understanding of metabolism in your organism, different organisms, and the sort of frontier of what enzymes are capable of, all of which are kind of advancing forward every year, this process of retrobiosynthesis is really often performed just using tacit or expert knowledge. But as we'll talk about soon, a long-standing goal in the field is to be able to use machines to be able to predict this. Not just to save people time, but to be able to explore more coherently, more logically, all the different candidate de novo pathways that one might want, that one might be able to test. And again, to really do that requires better cataloging of the biocatalysts that you have, and knowing that their substrate specificity on substrates that they don't naturally react on, and then also being able to potentially predict that, since those measurements have not all taken place. So that can be complicated. We'll come back to that. With our remaining time today, I just want to discuss a few examples briefly to provide some context for what are some de novo pathways that have been engineered, and why might you want to do this. So I thought some good examples here would include naturally occurring molecules, as well as some unnaturally occurring unnatural molecules. So here we have the de novo vanillin biosynthesis in plants. And so by this use of de novo, we mean this is how plants make vanillin from scratch. This is a pathway to vanillin that they're able to connect all the way back to simple inputs. But this is, a naturally occurring pathway. And what you can see about it, so first of all, there's multiple routes here. So plants have a few different ways of getting to vanillin, and one thing that appears to be in common for all of these routes is that they're downstream of phenylalanine. And so if we revisit aromatic amino acid biosynthesis, which we've looked at in homework and earlier lectures, we know that phenylalanine is pretty far downstream in this pathway that starts with DHAP and makes its way down to shikimate and then to phenylalanine. And so then we have potentially all these other transformations where phenylalanine and trans-cinnamic acid, and several of these metabolites all have a longer carbon side chain off of the aryl ring before it's essentially cleaved to form vanillin. And so this type of pathway might not be the most efficient. And as we think about pathways that were constructed experimentally, which are what these examples are going to showcase, we can see that there's opportunity both really from experimentalists as well as for computationalists to design more efficient routes using de novo pathway design. And so vanillin biosynthesis is exactly that kind of example. When it was shown in yeast, and in this case, de novo actually also refers to from glucose, but it is both a de novo, it's a de novo pathway in both senses. It is a novel route, and it enables vanillin formation from glucose. And that's actually important to highlight because a lot of vanillin could be made through biocatalysis from lignin or other kinds of intermediates. But this is the design of a pathway in an organism, in yeast, where it can generate vanillin, molecule 4 here, directly from sugar. And what's really neat about this biosynthetic route to get to species 4 is that you actually only need 3 heterologous enzymes. You can see this here. The OMT and the ACAR are just the same as here. And this might look familiar to the example that I brought up when we were talking about vanillate biosensors, because this is essentially the same pathway. The developers of this pathway in yeast designed a new pathway and demonstrated the production of vanillin from this metabolite in the shikimate pathway, dehydroshikimate. And it's relatively early in the aromatic amino acid biosynthesis pathway, which likely makes this route more efficient. It then was the same pathway that I took and put into E. coli. So there was relatively little innovation there, at least in terms of that part of what I was working on. But this was really neat. And here it actually shows further a step involving a glycosyltransferase to create the glucoside of vanillin for different reasons. But so this is meant to highlight an example of when de novo pathway design can be more useful than what nature does. And another area that I'm just going to mention has to do with the idea of functional groups that are not very common, and that includes these terminal alkynes and the molecules containing them. And so this type of approach towards terminal alkynes has really been pioneered by 2 labs at Berkeley. Those labs are Wenjun Zhang and Michelle Chang. And these are important natural products, but there's a lot of interest in the terminal alkyne for click chemistry. And so being able to figure out how to make those pathways and then to be able to engineer new pathways to other products that might contain a terminal alkyne is of interest to a lot of folks. And so the example that I just showed before, particularly this Jamaikamide B compound, is from the Zhang lab at Berkeley. They focused on its biosynthesis. Another example, this is from Michelle Chang's lab, was trying to elucidate the pathway to this amino acid that contains a terminal alkyne. Essentially, what both approaches utilized was this focus first on these are naturally occurring metabolites, but we have to figure out what the pathway exactly is first. And so in this kind of de novo biosynthesis examples, Here, it was maybe less pathway design than pathway elucidation. But the idea then is that the knowledge of how these natural pathways work will be the basis for future de novo pathway design. And so here, a lot of this work involves LCMS in order to figure out what are the intermediates. And you can see that here in this case too. And at least in the case of the work based on Jamaica-mide B, there was then follow-up work where the ability of the terminal alkyne to participate in click chemistry was used to click on fluorophores and then do enzyme engineering in order to improve the enzyme that converts this terminal alkene to a terminal alkyne. And so that's pretty neat. You can imagine that you wouldn't necessarily even just improve it on its native substrate, but that you could engineer it to have a broader substrate specificity to be able to convert other terminal alkenes to terminal alkynes.